My name is Arsen Hunanyan, and I am a postdoctoral research associate in Dr. Mikatis' lab. And I am doing basic science research, especially, particularly I am doing in mutated mice, which have two type of mutation. One is D801N, and another one E815K mutation. This is two different type of mutations. So we have these mice, and my research is to study what's happening during this mutation. First of all, in order to understand what's happening in patients, it is extremely important to have animal model of this disease, because you cannot do experiment on patients, but you can do a lot of experiment on mice. And so we created these mice. First of all, we are trying now to understand what is the abnormalities in a brain, some particular brain structure, to find any difference which is abnormal compared with normal mice. So far what we found that there is these mice, first of all, these mice have very close abnormalities like humans, and electrophysiological experiments, which is the experiments to measure the electrical activity of neurons in the brain networks, it revealed that there is some hyperexcitable neurons which are located in the hippocampus. And the hyperexcitable, it means that their electrical activity is higher compared with normal case. However, it is extremely important to mention also that even though we found this phenomena, first, to making this hyperexcitability less, it doesn't guarantee that you can help alternative hemophilia children, because it is very complex. It is not so simple. And if it's so complex, it requires too many research to find out all these abnormalities and to find out all brain connections which have connected with each other, and as an outcome can give many different abnormalities like in humans. It is very well known that it's involved in seizure. So if if we depress the abnormality of the hippocampus, there is a very big chance that we can depress the seizure activity. However, whether this is related with, let's say, hemiplegic episodes, we don't know. As I said, it is very complicated. It's not just simple, and of course it cannot be simple, because the mutation, it's not only in one cells, or it's all over the body, and it's in eye cells, in different brain cells, in the muscles, everywhere. So, but the way how science is developed and how we can help, it's to target one structure first, to investigate it deeply, and then to give it treatments to see whether it helps in the mouse, and then to go to the next step. If this particular treatment will help, let's say, seizure, but it will not help alternative hemiplegia episodes, then it means there is another structure involved. We have to go in that structure, and if we are smart enough, we can find this whole structure and eventually to develop the treatments which can hopefully help alternative hemiplegia of childhood. Of course, I'm optimistic, otherwise, I will not be here. We have to understand one thing, that to find a treatment which is the cause of mutation, it's extremely difficult. You can spend all your life and will find only a few things. But again, if God will help you also, you probably will find. It is not 1 year, 2 year scientific research, it's many, many years. And it's not only one scientific group, it's worldwide task. So, but if you are keeping working in one specific problem, sooner or later, I think the treatment will be found. I met actually with patients because for me it is extremely important to see whether our mouse, which have similar abnormalities like patients. This is one reason why it is important to meet with them. And the second, looks like that the patients is mainly children, and I also have children, and to find treatment will be very important because they are very small. They are children, and they also want to live in a healthy manner. They also want to have family, and to live how we live. So, and it is very for me. Because they are children, it will be double efforts to help to find a treatment to help them. I just want to say that spending time in the lab, I'm doing my best to find the abnormalities in the mouse model, which will eventually lead to develop a treatment for children. And it doesn't matter I'm in AHC field or in other field, this is my character, that if I am doing something, I have to find and I have to nail it down. This is, I think without this, it's impossible to find something. You have to spend a lot of time. You don't want to go home earlier or something like that. You have to work very hard, because if the disease is tough, you have to be tough in order to find the treatment.